Aeschbacher, and I'm going to talk to you about the genome-wide signal of selection against gene flow and how we can detect it. This is joint work that I did at UC Davis with Graham Coop. In the case where you have spatially varying selection, there may be certain alleles which are maladapted to certain patches or to certain genetic backgrounds. Others are particularly well adapted, and that may lead in the long term to local adaptation, even in the face of strong gene flow. It's well established that situations like this lead to an effective reduction in gene flow, especially at the focal sites where selection acts, but this effect can spill over to linked neutral sites and also to linked selected sites. And as we move along the genome, we expect to see a heterogeneity in differentiation between populations. And this is not a new idea. It has been explored empirically as well, especially by the group of Michael Nachman at Berkeley. So back in the early 80s, Petrie, Benson, and others came up with the concept of a reduced effective migration rate. So here I'm showing you the formula by Petrie, which is just the actual migration rate multiplied by a factor that depends on the selection coefficient and the recombination rate. Plotted against recombination rate, you can see that if you're closely linked to the selected site, the effective migration rate is reduced and it recovers as you increase the recombination distance from the selected site. In the late 90s then, Magnus Norborg, Deborah and Brian Charlesworth showed or derived the effect on pairwise coalescence time within and between populations of neutral sites that are linked to selected sites. And they also advocated the use of absolute divergence between populations as a measure rather than FST, because FST is confounded and several modes of selection can give the same signal in terms of FST, whereas absolute divergence can distinguish between these types of selection. So as a first motivating example, I'm going to use data by Yaniv Brandwein and Amanda Kenny. This paper is going to come out in the next couple of days. They have looked at integration from Mimulus nasutus into the sister species Mimulus guttatus, and they have found that if they plot the substitutions on the y-axis as a function of the local recombination rate on the x-axis, they found a significant negative correlation. So that could be evidence for selection against gene flow into the wrong genetic background. As a second example, I'm going to zoom in on Mimulus guttatus, which is known to have locally adapted to serpentine soil. Serpentine soil has a strong effect on plant communities because of heavy metals and iron concentrations that are usually toxic to plants. So Jessica Selby from John Willis' lab has planted serpentine seedlings on serpentine soil and non-serpentine seedlings on serpentine soil, and you can see only the serpentine seedlings germinated here. So it is evidence for a relatively strong local adaptation here. Again, if we plot between population divergence as a function of recombination rate by base pair, we see this significantly negative correlation. Again, that could suggest that there is selection against maladapted immigrants from non-serpentine patches. So to model this, we can imagine two populations which are connected by migration. For simplicity, I assume one-way migration here. And at some point in the past, at time tau, a selectively beneficial allele arose in the focal population. And then we can look at a closely linked neutral site and follow the trajectory of two lineages, one lineage from the focal site and one lineage from the source population, and can follow them back in time until they coalesce. So that guy is in the focal population for quite some time, then migrates back and then has a chance of coalescing. If we look at another locus which is more loosely linked, that one might actually recombine off the beneficial background and then migrate back into the population, into the source population, and then coalesce here. So to simplify this, we assume that there are two phases. So in the first period, we have a reduced effective migration rate, basically up to time tau, where the selectively beneficial allele arose. And we're going to model this by this term, so that's a chance of migrating by time t, and then we have a coalescent time of t plus the within population coalescent time in the source population, and we're integrating over t from 0 to tau. Then there's a second part. If we don't migrate in this phase here, we have a chance of migrating here, and we finally do so, and that will lead to this term here. So that's for a given selected locus at a given distance from the neutral site. But we want to integrate this over the genome. So we have a distribution function for the distance between the neutral site and the selected site, and we can integrate the previous expression over the whole length of the genome. And that will result in a function which I just abbreviate by G here because we don't have the analytically closed form so far, but it's going to be a function of all these parameters here, so effective population size, migration rate, selection coefficient, and here the rate at which we have neutral selected sites along the genome, and then importantly the per-base pair recombination rate. That's an important parameter because that's something we're able to measure, right? If you have a linkage map, we have access to these local recombination rates. Then if you multiply this expression by 2 times the mutation rate, we basically have an approximation to the between population diversity. So I'm going to call that pi the remaining part of the talk. So we have done some simulations using SLIM, biofield measure, a modified version actually. And what I'm plotting here is the pi as a function of the log of the recombination rate per base pair. And so you see the lines, the curves here, they correspond to the formula I showed you on the previous slide. And the dots and whiskers, they are from simulations. And you can see that we are doing an okay-ish job. It's not perfect here, the match, but yeah, that's because of drift playing a role in these simulations. We have a relatively small population size here, for instance. So from orange to blue, we are increasing the age of the beneficial mutation from 2,000 to 16,000 generations. And you can see in regions of high recombination, we basically reach the baseline that we would expect if the migration rate was equal to the absolute or original migration rate. And as we move closer to the selected site, then the between population diversity is increased. So we want to look at a number of selected loci, not just one. And so recently, with Reinhard Berger, I derived an approximation to the effective migration rate at the neutral site that is linked to an arbitrary number of selected sites on the left and right. So we can again compare the performance of this formula against simulations. The curves correspond to this formula here. I've added a bit of noise by choosing the selection coefficient from an exponential distribution and also choosing the distance between selected sites from an exponential distribution along the genome. The diamonds correspond to simulations. Again, I used SLIM. You can see the details here. And you can see for a weak selection, we have a component of drift here that is present in the simulations, but not in our theory. So that leads to deviation here. And for strong selection, we have the effect that multiple selected sites start interacting mutually. That's another effect we don't model currently in our theory. So we have a bad fit for drift-dominated scenarios and a bad fit, relatively bad fit, in scenarios where selected loci interact. But overall, the point I want to make here is that we pretty much model the decay very well. We have a deviation in the intercept. So we can further approximate that formula, the complicated formula that I showed you on the previous slide, by making a couple of assumptions, and that leads to a nice form. It basically says the effective migration rate is a product of the actual migration rate times an exponential function. And so in this exponential, in the exponent, we have the number of selected loci, and we have a constant, Euler's constant, which is about 0.6. And then we have this important compound parameter, which I call eta here, which is the product of the mean selection coefficient times the rate at which we place selected loci along the genome, divided by the local recombination rate. And so here is the comparison between this analytic approximation here, the black dotted line or dashed line, and I'm comparing it here to the previous formula, the more complicated formula, which I have computed here for 3 different scenarios. You can't see probably the different lines because they overlap perfectly, and that exactly illustrates the existence of this compound parameter. You can see that also the exponential form holds more or less, especially in the range where eta is small, and that's actually where we expect the approximation to hold. So, as an application, we come back to the Miolus cotatus example I showed you before. We have zoomed in on 2 populations, a serpentine population from the McLaughlin Research Station in the North Coast Range in California, and another population of serpentine about 30 miles south. We have obtained genome-wide sequence data from the Willis lab. We have aligned the reads to the reference genome 2, called SNPs, done a couple of standard filtering and correction steps. We've also obtained a physical linkage map from Lex Flegel and Yaniv Brandwein. And then we have pinned the data into windows of 100 KB. And then, basically using the formula I've shown you before, we can do a couple of algebraic modifications. So we have on the left our y-axis, basically, or our the variable we want to explain. That's going to be a function of the observed pi of the estimated population size. We estimate that from within population diversity. And on the right side, we have an intercept, which I call A here, that is related to the migration rate, and we have a 1 over the recombination rate per base pair. Remember, RBP is something we can measure from data. And then we have P, which is basically the slope, and that's going to be this compound parameter. So we have factored out the RBP from our previous parameter eta here. And so that's what we get if you fit this model to the data from Mimulus Cotatus. Again, on the y-axis you have the between pi, and on the x-axis you have recombination rate on a log scale each. And you see we explain about 22% of the variance. And then looking at the estimates, so our A, the intercept, is estimated to 7.53, and that translates into a migration rate of about 5 times 10 to the minus 4, so it's quite low. But remember, these populations are far apart, and also these low migration rate could just reflect other effects of genome-wide reduction in gene flow due to selection. But the more interesting part here is this compound parameter, which we estimate to this magic number of 1.5 times 10 to the minus 6. So remember, it's a compound parameter, so we can't really resolve that, but we can make an example, for instance. So we can assume that perhaps there were 200 selected loci that have an effect, and the average distance between these loci is, say, 100 kb, and that would correspond to each of these loci having an effect of 1% on average. So that's just an example of how this compound parameter could come into existence. So I've assumed a mutation rate of 10 to the minus 8 here, just so that you get an idea of the order of magnitude here. Alright, so I think I've shown you that selection against gene flow produces a unique genomic pattern, namely a negative correlation between population pi and local recombination rate. We have identified a compound parameter which basically quantifies the per base pair effect of selection against gene flow. So it's the product of the average selection coefficient times the rate at which we expect selected sites divided by the local recombination rate. And then last, we have quantified the aggregate effect of selection against migrants to serpentine patches in Mimulus Cotatus. So obviously people are interested in identifying individual loci that are subject to migration selection balance. Our approach can't do that. On the flip side, however, we have avoided all the issues that come with these outlier tests. So we don't have to deal with false positives. We are robust against demography. We can distinguish different modes of selection. And we are borrowing strength across multiple loci that contribute to local adaptation. And so if you think of a large number of small effect loci, you would be likely not to find them in an outlier test, just because they fail to be significant. But our method actually borrows strength across these loci, because we have this aggregate measured across the genome. Alright, so in the future we want to extend that model to allow for migration in both sites. We want to account for the interplay among selected loci, which I mentioned at the very beginning. We want to incorporate recent local sweeps and background selection, and of course essentially distinguish between these scenarios. And on the practical side, we are going to analyze many more pairs of serpentine and off-serpentine populations in Mimulus Cotatus along the west coast of North America. So then I want to thank you for listening. I want to thank the amazing Coop Lab at UC Davis, especially Graham and Chen Ling, who have been working together with me on that project. I want to thank Jessica and John Willis from Duke here, and various other people for help, discussion, or for provision of data. Thank you very much. Yeah, of course,